Hello, everyone, and welcome to Talk Nerdy. It's Monday, August 7th, and I'm your host, Cara Santamaria. And this week, we're going to be talking with the author of The Cell, Discovering the Microscopic World That Determines Our Health, Our Consciousness, and Our Future, Dr. Joshua Rappoport. But before we do, I want to thank those of you who have made Talk Nerdy possible this week. It's because of your ongoing support that I can continue to ensure that Talk Nerdy is free for everybody who wants to listen, 100% free to download. And, you know, it's expensive and it's time consuming to do this podcast all by myself. I book the episodes, I do all the recording, I do the uploading, the editing, which is why it's not always perfect. And also, you know, that requires sometimes travel. It requires good equipment. I also run the online store. And because of the support from you guys, I was able to get a full travel rig together that's going to sound really, really good now when I go out of town for conferences and conventions. I'll be able to interview really interesting people that I meet along the way, and it'll be just as good a quality as my in-studio recordings like the one I have today. So I want to thank you guys for that, and I want to give specific shout-outs to those of you who are my biggest supporters this week on PayPal, which you can visit by going to carasantamaria.com and clicking through to the PayPal link. That's where you make a one-time supportive payment. I want to thank Rob Schreck. And for my Patreon supporters, that's at patreon.com/talknerdy. I want to thank Ernesto Herrera Bustamante. I want to thank Phil T-Bear, and I want to thank Timothy Glover. They are supporting the show at the highest level. And then I also have a bunch of people supporting the show at the next tier, which is Genji Keene, Charles Payette, Jonathan Wright, Christian Jeffrey, Jeffrey Perez, Stuart Ogue, The Honorable Husband, Jafe, Gabriel Felipe Jaramillo Gonzalez, Brian Holden, and Jeffrey Sewell. Thank you guys from the bottom of my heart. And to the literally hundreds more of you that support at a different level, I thank you so much as well. Remember, there's other ways to support the show, like rating and reviewing it on iTunes or wherever you download your podcast, by sharing it via social media, and by visiting the Talk Nerdy store, where I now have all t-shirts in stock, as well as a lot of cool goodies like hand-thrown mugs that say Talk Nerdy, hand-tooled bookmarks and keychains with the Talk Nerdy logo, as well as just some fun stuff like stickers and buttons and slap bracelets. Oh, and also signed copies of Charlie and the Tortoise, both by the author, MJ Mouton, and by myself. I wrote the foreword for that book. So that's a great gift if you have some kiddos in your life. Hit me up and I'll send a signed copy out to you. And remember, if you want to go to the Talk Nerdy store and you don't live in America, you'll see that the shopping cart software doesn't support you. All you have to do is send me an email at shoptalknerdy@gmail.com and I'll be able to send you an invoice. I can send to anywhere in the world, so don't be deterred. All right, guys. I'm really excited about this week's guest. I mentioned previously, his name is Dr. Joshua Rappoport, and he is the director of the Center for Advanced Microscopy and Nikon Imaging Center at Northwestern University School of Medicine. What a freaking cool job. And he's going to teach us all about The Cell. He wrote a wonderful book that's now on paperback, and you can pick it up anywhere books are sold. It's called The Cell, Discovering the Microscopic World That Determines Our Health, Our Consciousness, and Our Future. So without any further ado, here he is, Dr. Joshua Rappoport. Well, Josh, thank you so much for joining me today.
It's a real pleasure. Thanks for inviting me.
Of course. So you have a new book out. It's got a long subtitle, so I'm going to read it. The Cell, Discovering the Microscopic World That Determines Our Health, Our Consciousness, and Our Future. And this was published in March of this year, right?
Yeah, I think it came out in April technically.
Oh,
yeah.
gotcha.
And
okay.
I just call it The Cell.
The Cell. Yeah, exactly. And so The Cell is a pretty big topic to dive into,
Yeah.
obviously. Are you a cell biologist? Like, what made you want to write about the cell?
Right. So I am a cell biologist. I mean, I guess you could say I'm a molecular cell biologist. I mean, that's at least the department I'm in. But I guess I look at the cell sort of as like a microcosm, or, you know, right, the definition is like the smallest thing that can be called life is the cell. So, you know, you can, from a reductionist perspective, you can look at the cell and learn a lot about, you know, health and disease and everything in biology.
Absolutely, because there are whole organisms that are only made up of one cell,
Exactly.
right? But they've got all the necessary molecular machines to be able to thrive. I guess we'll wait to talk about viruses and whether or not they're alive, because that's a whole complicated thing. But you got your PhD at Mount Sinai School of Medicine, actually in the biological sciences department at NYU. But your PhD is actually in, let's see, mechanisms of disease and therapeutics.
Right. So that was just kind of like I think a catch-all multidisciplinary title for the program, because you could do a lot of things in the program. My PhD supervisor was actually an MD, like a proper real doctor, but she had a basic science lab as well. So they wanted the program to have a sort of semi-translational, clinically relevant flair.
Sure.
So, yeah, but I've become very basic science and, as I said, reductionistic. And, I spend a lot of time looking at, cells and microscopes, and it's somewhat directly relevant to, biomedical topics, the research that I've done. But generally speaking, since my PhD, I've sort of gone in the more molecular, cellular, rather than physiological, medical area.
So do you then focus solely on human tissue, or do you look at cells across all different species?
Right. So generally speaking, I've focused on mammalian. So the cells are mostly going to be cell lines. So that means that they can sort of grow in perpetuity. We call them immortal or immortalized, which means they'll just keep replicating over and over again. So a lot of these are either cancer cell lines, or they've had some kind of a mutation or a virus infection that has allowed them to just keep reproducing. And so they've sort of lost a lot of specific phenotypic characteristics, but they're a really useful tool. And it sort of, at some level, it doesn't really matter if they're mouse or they're rat or they're, human from origin.
Gotcha. Do you work with HeLa cells?
Absolutely, yeah.
Yeah, so
Absolutely,
I feel like
100%.
people might actually finally be more aware of what that is, because obviously Rebecca Skloot's book, I think, was super popular. A lot of people might have read The Immortal Life of Henrietta Lacks, but then they made it into this great HBO film. Did you see it?
So I read the book a couple of years ago, and I felt the book was amazing, and the descriptions were really great. And I think in writing my book, I sort of assumed that a lot of people who would read my book had read that book. And so I didn't go into huge detail about, what it means to have a cell that, can grow in culture and that kind of stuff. So, I think the HBO thing, I mean, just my own personal, I found it sort of they didn't include as much of the real sort of hard science that was in the book. So it was more of a interpersonal story than it was the nonfiction science perspective.
Absolutely. Like Rebecca's book, I think, was sort of ran those two stories in parallel. It was the story of the family of Henrietta and the story of her cells being taken without her permission, and just medical ethics questions around the era that they were first taken. But also there was a lot of very good science, I think, teaching people about the basics of cell biology and about cancer biology. And of course, you're right. The HBO movie, it was only an hour and a half. They focused on the story a little bit more than the science. They kind of just barfed all that out in the first 5 minutes. Like, okay, this is what it is. This is what it means.
But it was a drama. It wasn't like a documentary or something.
Absolutely. Yeah. So that's cool. That's cool to know that you use these tools that I think I always love in science communication when we talk about certain types of research, and we can dive deep, like in a podcast like this, how we can connect it to other areas of science communication so that people who are listening, who are interested, who maybe don't have any background in biology, can say, oh my gosh, I can make these connections now. Like, I read that book, and these are the cells that he's using when he does his research. So that's really cool.
I mean, I think it's useful as a science communicator, also for us to look at really good examples and sometimes not so good examples of, cautionary tales, or to, follow the path. I know my publisher, when I first pitched the idea, he had me read a book called The Lives of the Cell, which is like from the 60s. It was written by...
I think I have that book.
It's a great book. It was written by like a retired MD, and what he had me read it for was not really the information contained within it, most of which really is sort of out of date, but still relevant to understand historically. But because it was written as a series of sort of independent essays that were sort of clustered together, and it does a great job of linking from one to the other. And so he really showed me that as an example, and I really used that as a sort of guiding principle in how I organize the book. So
that's great.
It's not like it's not, as much as I wish it were, it's not the kind of book where like you can't put it down, you sort of get to the end of a chapter and you're like, okay, I can walk away from it now. But there will be a transition, hopefully, to the next chapter, but it really is more of like a series of self-contained essays.
I like that, though. I feel like both of those approaches are, to me at least, equally important, because sometimes I want a book that's got a narrative arc that's like a huge page turner, right? Mm. But sometimes I want a book where I can read one chapter every night before I go to bed and feel like it's really self-contained. And later on, if I want to reread the book or I want to pick it up, I can just go to a random chapter and read it, and I don't have to preface it, with a bunch beforehand.
Yeah, I think like the Hot Zone is another really good example of a nonfiction science book that has a narrative flow.
Yeah.
And it sort of takes little tangents and will give you a good deal of information, but then it's sort of following this harrowing tale.
Yeah.
Whereas I think mine, like you could be like, oh wait, I want to learn more about X, and you could find the chapter of that and then look it up and
Absolutely.
it can stand alone.
Yeah. We've been working on a book for the other podcast that I do, The Skeptic's Guide to the Universe. I think we just announced maybe like a month ago that we've finished the first major draft, got it to the publisher, so now we're in that long process. But ours is written much like that as well. It's these sort of vignetted chapters about, this skeptical principle, that skeptical. It's a guide. So you can really dive into it at any major point. See, now I'm like scanning my bookshelf to see if I have that book. I'm gonna have to look afterwards and make sure that it's the same. It was written in like the 60s. Oh, The Lives of the Cell by Lewis Thomas. Ha! Hilarious. I love it. Okay.
But I think, one thing that I really tried to do is not feel the stress of trying to be comprehensive.
Yeah.
So I sort of, when I wrote the book, I sort of asked myself what's really interesting, what has stuck with me over time, and what do I think others would find interesting and accessible is the other thing. And it was actually, I wrote it pretty quickly because I felt like I didn't have to do I didn't feel like, oh, there's this topic that I need to include, so I'm going to have to do a bunch of research and I'm going to have to understand it and then I'm going to have to write about it. It was more like stuff that I had already encountered and what I wanted to share, express to others, and, do it in a comprehensible way. And so it actually kind of all just came out of me. There were one or two chapters that the publishers were like, you should probably include a chapter on X because there seems to be a gap there.
Yeah.
But generally speaking, it's a bit of a mixed bag, sort of heterogeneous group of things that I find particularly interesting. That's really the common theme throughout it.
I love that. And it's not a textbook. Like, this is not a book where you just go chapter by chapter, like explaining the basic mechanisms of a cell. Like, there's some of that background in it, and that's really important. But there's a lot of really cool stuff in here about, what's happening in a cell, why a cell really is that basic mechanism of life, the genetic information contained within a cell, like what is a nucleus, how is DNA organized, why does it matter, But you also dive into some topics that I think are so important for science communication. So I think before we even actually get into what a cell is, why a cell matters, why, for example, you've been willing and fascinated to dedicate your entire career into this basic unit of life, you've also talked a bit about science communication, basically, about science journalism more specifically, and sort of the difference between good coverage, legitimate coverage that tells the story of the science in a responsible way, and sort of, bad side journalism, which we all have to encounter all the time. And I feel like it's one of the most common questions, at least in the emails that we get for the Skeptic's Guide. Like, how do you know when you're just reading popular science what's garbage and what's not? Why did you feel the need to include some of those conversations in your book?
Well, I think people who are going to be interested in learning more about science, there are always going to be those things that stick out like a sore thumb. Like I mean, I don't want to specifically badmouth anything, but let's say like homeopathic remedies or something.
Absolutely. Yeah.
Things that just, if you're at all skeptical, if you're all empirical, if you're looking for real evidence, you're going to just discount that. And that's one thing. And there are clearly people that can be fooled into, and I think it's really important because, if you're fooled into some, snake oil, and you don't go to a proper doctor, really bad things can happen. It's one thing if you're like, oh, I want some kind of tonic to make me feel better before, I go to the gym or whatever. But if it's actually a question of therapy for some disease or problem, then that can be really serious. And I think that's one side of it. But almost to me, more relevant to the audience would be this question, of how do you know good coverage from sort of sketchy coverage, and I do, I feel like for example, communications departments in universities. Like I think the people are genuinely trying to do their job as well as they can, but they're more in marketing than they are in science communication.
Absolutely.
Really, really, it's about getting the university's name picked up by newspapers and magazines, and really having press releases from that point of view, as opposed to really, I don't want to say honest, but accurate communication of what the actual science is. And you read a lot. I mean, I'm kind of I'm sort of like a closet crank. Like I've written several emails to science authors from major newspapers and said, actually, I've done research in this area, and the way you expressed it was inaccurate or incomplete or whatever. And there are 3 or 4 times when articles have actually changed because I read them online, and they've actually changed them because of that.
That's great.
So, but I do it totally like, I'm not doing it like in the comments section, or whatever. I email usually you can find, if you try it, you can find the author's, email. And usually the authors or the journalists are happy to hear that.
Absolutely.
So it really, it's sort of like, effective communication. What does that mean? Does it mean, and I think it means it's accessible and accurate. It doesn't mean making people say, wow, that's amazing. There are all these like memes about, isn't this amazing? So, I love science, blah, blah, blah. And really, science isn't the eureka moments and the, one composite, cartoon that came out of, hundreds and hundreds of hours of work. It's the painstaking time that people spend behind the bench, usually dealing with frustration and failure.
And it's so incremental. And that's the really, I think, frustrating part for a lot of people, because science news moves quite slowly. But journalists who are tasked with trying to make splashy stories on a daily or a weekly basis really have a difficult time, right? And so sometimes, I think things are made to be a little bit more conclusive than they are.
And so I've sort of been on the other side of it and been the scientist who's been interviewed about the science. And, I've tried to be as accurate as possible about the incremental nature and about, but I'm still excited about it. I mean, obviously this is what I do, so I get excited about it. But the person doing the interviewing can be sort of like, okay, but what could potentially this mean, possibly years in the future kind of a thing? Because that's the catch. That's the headline.
It's interesting how basic research, and it's like you get it, right? Like you put yourself in the shoes of the journalist, and it's like, how do I sell a story to a general audience about basic research versus what's the word I'm looking for?
Applied,
versus applied research. And how do I make that story have a human focus? How do I make it feel relevant to somebody's life? It's the same reason that I think personally chemistry gets short shrift in science journalism. You just don't hear chemistry stories that often. You hear a lot of physics stories, these huge big picture cosmology, because then it can be like really philosophical in nature. And then you hear cancer biology, Alzheimer's research, Parkinson's research, a lot of things that not just that they're splashy and they're sexy, but that real people who don't have a strong background in science can read about and go, oh, crap, that could help me. That could help my family. That's why I'm interested in it. Because we're, narcissistic by nature. This is not a commentary on the public. This is a commentary on being human, right? Like the best science communication, as many people know, the reason that Rebecca Skloot's book was so powerful is because they focus on human stories. And so it is, I always try to see it from both sides, having worked as a scientist and having worked as a journalist, to try and kind of understand what the drivers are. And then it's even that much more obvious when egregious mistakes are made in science coverage, because sometimes it's so unethical the way that these experiments are covered. This is what was found in this study. These are the limitations. These are the questions. Oh, and by the way, they're going to cure cancer now. Yeah, it's like, oh, Jesus Christ.
I think, though, also as a scientist, you're sort of, you want to self-promote, right? You want to get your name out there. Yeah. You want, there are lots of scientists who have Twitter accounts and they're tweeting, oh, we just had a paper published in Nature, blah, blah, blah.
Yeah, of course.
That is sort of part of it, which seems a bit strange.
It's hard. And there's a push and pull between, I think, the old guard and the new guard, because it does feel like a lot of younger, my dog is like really stressed out for some reason. Killer, there's construction outside. It's not for you. It's not for you. Because a lot of, I think, younger younger scientists are becoming more interested in the concept of science communication. And so it does seem like they're learning the tenets and they're learning how to find balance in those approaches. Whereas with the old guard, it's like extremes. It's either like never self-promote, never talk about your science to the public or to journalists, because they'll always butcher it. And you should just keep your head down and focus on the science, and the mistakes they make are theirs. Or like, yeah, I'm going to send out a press release because I won this award, which is, I think fine. I'm not judging that, but finding a good balance. I think, it comes down to exactly what you're talking about. I think if grad students are trained in science communication, they're going to be able to approach it in a more balanced, more ethical kind of way.
Yeah, but I think at the end of the day, and this sort of brings it back to the book, is really what science is about. And like you said, it's incremental. But you know what? We're now decades and decades into what I would call the sort of, modern biomedical, world.
Absolutely.
And so there's a lot of information out there, and you can just, look, for example, if you want to talk about vaccines, there's a lot of information out there, and it's not an incremental thing that needs to, that either you're going to, self-promote. It's a question of communicating really important information that is cumulative and kind of unequivocal. Same thing. I mean, it's not my area, but the same thing would go with climate change, right?
Yeah.
It's just like it's the responsibility of scientists. I mean, so I had a professor in my undergrad at Brown. His name's Ken Miller, and he's like a cell biologist, electron microscopist type person, and he sort of took it upon himself to speak out in support of evolution being taught in schools when it's under threat and people are skeptical about natural selection as a theory and all that stuff.
Is this the same Ken Miller who's like a religious guy? But
well, so that's the thing. That's the thing.
I know him. I feel like I've had meetings with him.
Okay, so he's amazing.
Yeah.
And the thing is that what he's really known for is not his own area of research, but he sort of took this mantle of science communicator on because he taught intro bio at Brown. It was hundreds and hundreds of people, the biggest class I ever took. Mm-hmm. And he was the best lecturer by far. Like he could stand in front of a room of hundreds of students and get them all completely captivated. So he's a great communicator, just full stop. Yeah. And then he took it upon himself to then go and, sort of prosecute this scientific argument with regards to evolution, even though he's not a geneticist, he's not an evolutionary biologist.
And he's not an atheist. He's like a staunch Catholic, right? I think he's Catholic.
I don't know about staunch, but I know that he
Yeah.
can speak to it from a perspective of even though
which I think makes it more
recognize- yeah.
effective that way, obviously, because he can say, listen, like I'm with you in this, like I believe in God, I believe in these tenets of the religion, but that doesn't mean that creationism should be taught in schools. That's anti-scientific.
Yeah. I mean, it does get. I mean, I think it's. I don't want to go too far afield, but he is Catholic, and right there, the official stance, I think, and there's a lot of Jesuit science, and the official stance, I think, on the Catholic side is sort of that. Let's just say that I think the people that are on school boards that are saying evolution should not be taught in schools are mostly not Catholics.
No, absolutely not. I think that they're evangelical. Yeah, they're definitely, like, I'm from Texas. I see this all the time. Obviously, it's the Texas school board that's like making a lot of these decisions about textbook publishing, and these are the ones trying to put creation, Texas, Louisiana, Tennessee, like there's a lot of states in the South. But you're right, it's usually not Catholics. It's usually not even, what we think of as these more kind of traditional American denominations, Methodists and Episcopalians and things like that. It's like these staunch evangelical, maybe Southern Baptist groups like that. And it's a bummer. It's a bummer that we see that. We see this a lot in popular culture, this sort of manufactured fight between science and religion, when that's not really the dichotomy, right? The dichotomy is science and non-science, or science and ignorance, or science and, honestly, I think science and some sort of agenda-driven, political viewpoint.
Yeah, I think as a scientist, we're sort of, we're not, because we're generally logic-based and evidence-based, it's sort of impossible for most scientists to understand a perspective that isn't logic and evidence-based. And so when there's this talk of the other side of the argument, right? Like the other side of the argument still would have experiments and data points and statistical analysis. The other side of a scientific argument is not belief or gut feeling.
Yeah.
So, if you want to talk about like the other side of the argument for, safety of vaccines, or the evolution occurred the way, we think in a Darwinian sense, or, all these things, then you can look at experiments that, were done, or papers that were done, and, might, usually it's going to be this, it's like, with climate change, there is, nothing is ever completely cut and dry. And so, we talk about, confidence intervals or whatever when we're talking about statistical analysis. So nothing's ever absolutely 100%. And the fact that it's not absolutely 100% is a good thing, right? Because if it was, then whenever a grad student would show me data or whatever, and it would be too good. It always
it's like,
raises suspicion. So the fact that nothing is always 100%, it doesn't mean, oh, this is sort of like a place to stick the crowbar in and be not trusting the data.
Yeah, and dismantle all of it.
Because there's just inherent biological variability, right?
Yeah, 95% confident does not mean false,
Yeah,
right?
exactly.
It means pretty much true. Yeah. And I think that's sometimes a part of the conversation that we have to include as science communicators, is like, science by nature is a conservative venture. Scientists talk about their findings in a very conservative way. And I don't mean politically conservative. I mean, we're never going to say, this proves that. Or we're certain that, we always say things like the data show. Yeah.
Well, it's really this question of false equivalencies, right? Which is that, if you can say, oh, well, you're not 100% sure, that means it's equally possible that it's that it's not true. Like, no, we're greater than 95% sure that it's true. We're just not 100% sure that it's true.
Absolutely. Yeah. And I think that, we've talked about this on the show before, but John Oliver did that great segment on his show where he was talking about that false equivalence and that false balance in journalism between a climate denier and a climate scientist. And it's like, well, if 97% of climate scientists agree, then there should be 97 climate scientists sitting at the table. And those three, deniers will be drowned out by the actual data. I think it's really cool.
It does get really complicated though when you really want to. I think the real sort of gap that we have to bridge though, and the real issue is that when I have to review a manuscript, like a paper that's going to be published by a journal or a grant, it takes hours and hours to just go through a small bit of data and how they did the experiments and whether their conclusions are sound and stuff like that. And I'm only asked to review things that I'm an expert on, and it still takes me hours and hours to do it. So it's hard because you don't want science to be this sort of secret society with a secret language and just trust us, we know what we're doing kind of thing. But if I started to explain to somebody how we published a paper and it showed this, and this is why it's important, and this is how we came to that conclusion, their eyes are gonna glaze over. I mean, even most scientists' eyes are gonna glaze over.
Yeah, it's tough. It's tough because it's like science is not inscrutable. It's not, but it can often come across that way because in order to speak the language of a scientist, I mean, the reason that scientists can, like you said, review other scientists' work or communicate in a very high-level way is because you put in the 8, 10, 20 years of your life getting to that place where you are and even still, most scientists, at least young career scientists, are not that confident and have horrible imposter syndrome and are like, oh, I don't even know what I'm doing. So it's hard to expect that you can sort of simplify things down to these small bites. Oftentimes when I talk about science communication, one of the tenets that I really do hold near and dear to my heart is that we should never underestimate the intelligence of the audience, but we should always underestimate their vocabulary. Like there are a lot of things that conceptually, I think if we are creative in our presentation, the general public would have no problem understanding. Like these are basic human problems, basic logical problems. The problem is they're so, intertwined in a lot of jargon and in a lot of specifics. And sometimes I think for the story, the specifics might not matter. Sometimes they really, really matter because it's a question of if you approached it using this statistical analysis versus that statistical analysis, you got wildly different results. But sometimes some of those specifics aren't as relevant to the story. The problem, I think, and it seems like you sort of agree, I'm interested in your take, is that we've got to get back to a place where the idea of awe is fundamental. We don't have to invent things that are anti-science or invent things that are magical or mystical or beyond the natural world in order to, to find awe in nature. It exists that way. So telling the stories about how amazing science is or how amazing nature is at the most fundamental level, we've got to get back to a place where that captures the imagination of people without having to embellish.
I mean, I think that's why I got sort of bitten by the microscope bug. I mean, the things that we see, in particular, my area is live cell imaging. And so being able to basically have cells that are expressing inherently fluorescent proteins and multiple colors, and then you watch the cells perform some task or undergo some change, and you see the location of the proteins that you want to track. And awe is the exact right word. I think, I took a little bit of art history, and I think art history to me, having just really scratched the surface of it, is a little bit analogous, right? Because you can say like, oh, the Mona Lisa is amazing, or the Last Supper, or whatever. But like, an art historian can tell you for hours and hours and hours about the technique, about the materials, about the composition, about the use of light and all this stuff. But you just sort of stand there and, I mean, I know a lot of people just look at them and say, wow, because it's really famous, but there's a reason why it's famous.
Yeah.
And so there is this awe, if you actually see one of those things in person, one of those absolute classical masterpieces, and you don't need to listen to hours and hours of explanation of why it's amazing to sort of get that sense.
Yeah, you're right. And I think that your book is a perfect example of that because obviously it's called The Cell, but it's a manageable length. It's not overstuffed. Like I have seen other books on, I would say in university bookstores, textbooks that are also called The Cell that are 1,000 pages, just full of so much. And of course, that makes sense. You're teaching a student about the cell at the most fundamental level, and you want them to be prepared in their career as a cell biologist. This book is obviously for general consumption.
Exactly.
And the fact that you're able to tell this beautiful story of this most basic building block without beating it over people's heads and getting it down to the most nitty gritty. It's important to be able to do that.
And as I said before, I did not feel a responsibility to be comprehensive.
Yeah.
I felt a responsibility to be accurate and accessible to whoever might pick it up. And I mean, there are definitely things in there that other biologists or other scientists won't know or won't have thought about. But it is specifically directed at, anybody and everybody, really.
Absolutely.
And hopefully it is accessible. And, like it gave me some freedom to actually put some opinion in there.
Yeah.
When I do talk about these issues of false equivalences, and I do talk about the importance of sort of empirical evidence, and the risk of just accepting things at face value and not being skeptical, but also the onus that's put both on the expert as well as the sort of consumer. As I said, it is complicated stuff. And so you do need to think about it. You do need to pay attention to it. And we definitely live in a culture where, you get a sort of very short, blast of a vignette of something, and you're supposed to be able to take away from something from it. But you need a little bit more depth. But that I've tried to balance that by making, as I said, the chapters sort of individual chapters, standalone, short length, accessible, so you can get sort of one punchline in a couple of pages.
Absolutely. Oh, it's after my own heart for sure. All right, let's get back to the show. So you started to talk about microscopy. I would love to
Right.
dive into that a little bit more. You're talking about looking at cells under the microscope, a light microscope specifically, that picks up fluorescence. So when you talk about, because you said you use the term inherently fluorescent. So when you're looking at these cells, you're not adding things to them to make them fluoresce, or you are?
So there are lots of variations. So
Yeah.
first of all, there are obviously microscopes that don't involve any stain or any, you can just look at the inherent contrast in something. I mean, these are sort of the oldest type of microscope.
Yeah, like if you had one as a kid that had a little mirror and it would like reflect the light from your ceiling.
So you can look at like little, you can take pond water and look at little algae that are inside of the pond water or whatever.
Yep.
And then obviously an entire other world would be electron microscopes.
Yeah.
And those are not using light. They're not using photons. They're using electron beams, which are sort of give you super, super high resolution, very high magnification. But generally speaking, everything is sort of fixed in place and stained and, has been, coated or treated or whatever, and it's sort of a freeze frame kind of approach.
Yeah, you're not going to see an amoeba, moving around under an electron microscope. So these are the images that people might be familiar with of like a tardigrade in super high definition.
Yeah, like a pollen grain or whatever.
Or a dust mite looking like a monster.
Exactly.
Like that's,
But then when we're talking about fluorescence microscopes, right? So fluorescence, it's like, black light in your white shirt glows under a black light. So basically what you have is photons are absorbed by a material, and then that material emits a photon of a different color. And so you can detect those. And so things that do fluorescence efficiently and reproducibly, we call them fluorophores. And there are many fluorophores that are chemicals that we can link on to label But again, this would be in like a fixed cell, like it's been treated with formaldehyde or whatever. And then we add these labels. Usually we can add antibodies actually because they'll very specifically recognize some protein of interest. So if you want to know where that protein is, you get an antibody that recognizes it. And then you put a fluorophore onto that antibody, and then you stick it on, and then you can look at it in the microscope. But again, that's going to be a dead cell. It's going to be chemically treated.
This is like the kind of research that I did for a while with birds when we were trying to look at adult neurogenesis, right? New neurons showing up in the bird brain. You have to sacrifice the bird. You have to take little, you have to fix the brain and
cut
paraffin or whatever, cut it into slices, and then under the microscope, look for these markers. Oh, that's a new cell. That's not a new cell. That's a new cell, and you count, and you use the computer software to circle them.
And that's absolutely incredibly valuable. And so there's always going to be a trade-off, right? So the trade-off is that you, generally speaking, if you do the experiments right, that you're, really looking at the proteins that you're interested in or the markers you're interested in and in their sort of normal location and at their normal amounts kind of a thing.
Yeah.
But it's fixed and dead and sliced.
Absolutely.
And all
You're
that.
looking at a cross-section of time in the
So,
past.
whereas with live cell imaging, the trick is how do you label specifically the thing that you want to image, and how do you label it in a way that doesn't affect its location or function or expression level or whatever.
And so do you use proteins?
But it gives you the power of being able to watch the events unfold in real time. So there is, it all started with a jellyfish called Aquaria victoria, and this jellyfish is inherently fluorescent. And it has this thing called the green fluorescent protein inside of it. Now, this is a very long story short kind of explanation because it's actually much more complicated inside of the actual jellyfish. But suffice to say, it's been the gene that makes that green fluorescent protein has been taken out of the jellyfish, and it's been mutated a bit to make it sort of brighter and more predictable and stable. And then you can take that piece of DNA that will then make that green fluorescent protein, and you can attach it to the DNA that will make a protein that you want to study. And you do that in a way that there's no stop between the two. So it just becomes one protein, sort of that's half your protein of interest and half the GFP, green fluorescent protein. And usually this works on a molecular level, and usually it expresses in the right place at the right time at the right levels, and usually it's functional.
And so usually you can look under a microscope and you can see traces of this fluorescence in whatever you're intentionally trying to study.
Exactly.
They've also done this at like a more organismal level, right?
Absolutely.
Where you've seen the images probably online of like a glowing mouse or maybe a glowing kitten or something. It's because they've used GFP to stain skin cells or something like that.
And in the world of sort of molecular cell biology, there's this whole library of we call them DNA constructs that will have. I mean, you can buy them. They're also sort of free services that will bank them, and you can just get them from people if they've made them and published them. They should send them to you as a sort of shared reagent. And not just green. So the real power comes in when you add multiple colors. So you can look at multiple things relative to each other in space and time. So there are other organisms as well that are inherently fluorescent. And it's actually kind of cool because it's where sort of ecology really meets molecular cell biology. And there literally are people that are, swimming around in scuba masks looking for fluorescent sea organisms, for example.
Yeah, they often occur in the sea, don't they?
Yeah.
So like I call them my pets. I have millions of pets, these little dinoflagellates in a bottle next to my bed that are bioluminescent at night. So they glow blue. Yeah, and they're really beautiful.
And so luminescence is slightly different because with luminescence, usually it's some like enzymatic process that's going to consume energy, and a light will be released, as opposed to fluorescence where it's absorbed.
And you don't have to hit it with a higher wavelength of light.
In luminescence, yeah. So the most obvious example of the luminescence, right, is fireflies.
Oh, yeah.
Yeah. Cool. So the thing that the enzyme is called luciferase, and then there's a substrate called luciferin, and there's a chapter in the book about it.
Interesting.
That's an alternative to fluorescence is luminescence, and there are pros and cons depending on the experiment that you're doing.
But these fluorescent microscopes actually hit them with the right wavelength of light so that they glow for you.
Exactly.
And that's Isn't chlorophyll fluorescent to a
So,
certain extent?
so in fact, yeah. So we, as microscopists, we talk about autofluorescence, and it's
Okay.
usually a bad thing.
Oh, so
So
it
yeah,
could
so
get in the
if
way.
you want to image, well, there are lots of autofluorescent compounds actually, and different organs in our body, cells from different organs will have different levels of autofluorescence, which can make it
cool.
really difficult to image specifically in that same color, same channel. And plant cells have their own problems with autofluorescence as well.
Yeah, like the green chlorophyll will often fluoresce red.
That's right.
Underneath.
Yeah, that's
so
exactly right.
so I could see if you're trying to study that and you want to use a fluorescent molecule, right? Then you wouldn't want to pick something that fluoresces the same color because it would get complicated.
But usually if you're talking about a sort of purified prep of cells from a specific organ or specific organism or whatever, there'll be some kind of spectral signature of that autofluorescence, and we actually have microscopes where you can do a sort of spectral map, and then you can sort of subtract that out. So it's like, how you can take noise out of sound, out of
this
sound
audio
file.
recording.
Exactly. So it's like noise canceling headphones or whatever. So we can take an unstained, just shows the autofluorescence, record the spectrum of that, and then take the stained sample and subtract out the autofluorescent signal, so we can just image the signal that we want.
God, technology's cool. There's like coming up with workarounds for everything. So you were speaking about these inherently fluorescent organisms, for example, that then you're able to isolate certain proteins from.
They've taken them out of different organisms, and they're very good green ones. They're very good red ones.
How about blue?
So blue has been challenging,
Really?
actually, yeah. And there's some good what we call far red, which would be sort of near infrared, but they can be a little bit challenging to work with. So the real great thing about doing it with the antibodies and the chemical fluorophores is that you can do many colors in combination with real freedom, because if the spectra are distinct from each other, you can pick them out very easily. Whereas with the fluorescent proteins, you're usually, generally speaking, you're limited to 2 colors, maybe 3.
Gotcha. And so what are some of the processes? Like, is this sort of sky's the limit? You can really attach these proteins to almost anything within the cell?
For the most part, yeah, for the most part. And also, it's not even just, for example, it's not just, cell culture like I was explaining before. You can actually, as you said, you can get, transgenic organisms that will express them specifically in one cell type or another, and you can look for the emergence of that cell type, or the motion of that cell type, or the division or the death of that one cell type, because it's been specifically genetically marked.
Yeah, that's cool. So these organisms are actually either bred or cloned to be born this way.
Yeah, so you can do fruit fly brains, for example, and look at specific neuron populations. That's what my wife does, actually.
Yeah, that's neat. That's really neat. Oh, so she uses fluorescence in her research.
Yes, she does. Yeah, absolutely.
A little crossover there.
Exactly.
I love it.
Yeah, but she's a neurogeneticist, and I'm a cell biologist, so it is quite distinct.
Yeah.
But yeah.
But it is funny how a lot of these tools, especially the more sophisticated it feels like science has become, because we have so many years now of, like you said, this revolution actually happened quite a few years ago now. So we have all these tools at our disposal. The science is getting more, I think, sophisticated, more high level. It's almost impossible to stay siloed as a scientist, right? You have to kind of dip your toe into these other aspects of science and learn from them and utilize some of the techniques that they've developed.
I'd say sort of, it probably started before, but, since I did my PhD, I've seen a real shift for good and ill. From, this is my lab, this is the thing I'm interested in, and I'm going to get a grant to just look at this one thing, to now it's more like you're part of a team, and there's got to be a basic side of the team, and there's got to be a translational or applied side of the team, and there's got to be a a spin, and I don't mean spin like it has to be sort of, fake.
Yeah.
But I mean, there has to be an application. There has to be a sort of spin out. There has to be a rationale for what could be done with this.
Yeah, a lot of times that's linked to the funding, right? If you can't kind of show, if you're looking for federal sources of funding, if you can't show them that this is going to like affect people's lives for the better, it's hard to get money.
Exactly.
Yeah.
And the problem is that, and I say this in the book, actually, from my point of view, a lot of advances that have had the most impact, it's not like people were looking for that application. They were doing this sort of blue skies basic research. The person who looked at a green fluorescent jellyfish and was saying, "Oh, I wonder why this is fluorescent," didn't necessarily think, "Oh, and one day this is going to be a tool used, in every lab, used in, guiding surgery or whatever." It was just a question of being interested, probably.
Yeah, and it becomes difficult when it's hard to get enough sources of funding to be able to do these, to ask these basic scientific questions. So I think that some scientists have a workaround for that. Like you said, like they'll sort of have one component of the lab work that's more applied, but really they're still asking these fundamental questions. But then they have to kind of, money's fungible. They have to be really smart about how they use that grant funding. And it's so political, isn't it? Everything gets to be really political.
Yeah, I mean, I guess my own experience was, so I should say that I've sort of transitioned out of this world a little bit, because now, so the job that I have is like the most fun job, because
tell me more.
I run a microscope facility. So my staff and I have all these microscopes. Any one of them costs as much as like a house.
Yeah.
They all do different things, and we support them. We get to teach people how to use them properly, how to make sure they don't break them, and all the way through to how they design their experiment, what fluorophores they're going to use, what microscopes they're going to use, to how they're going to collect and then ultimately analyze the data.
Oh, neat.
So we get to be these sort of like paid collaborators almost, these sort of experts. And it's really not about my own research interests anymore. It's not my laboratory. It's a facility that we run for the Northwestern University Medical School, the university as a whole, Chicago, whatever. Like, people can come and use our microscopes, their hourly rates, you can schedule time, blah, blah, blah. And so it's this huge sense of relief, first of all, because I'm not constantly stressing about the next research grant or the next paper.
Yeah.
And I did not feel this, I don't really miss it. This sort of, being able to focus in on my specific research question. And I think what's happened is, organically, the people who are around, who have common research interests, they collaborate with me, and I'm still able to work on similar topics as I was when it was my lab.
Yeah.
But I'm just doing it as, not the senior author on the paper or not the PI on the grant.
That's really cool. And do you ever, you mentioned that you took some art history. Now you are working sort of in this shared service where you're providing these, providing the microscopes, but also providing the expertise and helping people, I think, get the best images for their research and analyze them the best way. Do you ever do kind of artistic microscopy? I know I used to judge, you probably know all about it, the photo micrography competition, the micro photo micrography competition.
Yeah, there
for
were
Nikon.
A bunch of them.
Like Small World or something.
Oh, yeah,
like that.
absolutely.
And
yeah.
it was so much fun to do these judges because you had all this criteria, like how important is this for, furthering scientific knowledge, but also like just how beautiful is it?
So,
what are we learning from it?
So in fact, the bulk of the research that my lab did, or I did when I was a postdoc or whatever, is really pushing the microscopes to a point where we were looking at sort of individual structures that were maybe 50, 200 nanometers across, like really, really small, which means that generally speaking, the stuff we were looking at was incredibly pixelated. And usually it was like single color, really fast live cell stuff. And it was completely inappropriate for those kinds of
Gotcha.
totally not artistic.
So
at
important
all.
scientifically, so
Yeah.
not pretty to the naked eye of the
You know,
public.
it's like it's like the raw data that's coming out of a radio telescope,
Yes.
right?
It's very different from a Hubble image.
Is very different from a Hubble image.
Yeah.
That's exactly right. And not that the Hubble images aren't important, but a lot of what I think astronomers and astrophysicists are looking at is really not going to be on the front page of a newspaper.
Absolutely. It's not going to be your desktop wallpaper. But do you ever, now that you're working in the shared facility, get to play a little bit and say, oh, this is really fun. Can I take a few hours to try and get a really beautiful image?
So first of all, there's still one or two systems that I'm the sort of point person on.
Mm-hmm.
So I do still get to train people and help people. I get to look at a lot of other people's data and advise them. What's always the worst is when you say, oh, you have to do it again because, the images were all saturated or whatever.
Yeah.
That's, not good for anybody, but it's a responsibility and it's sort of what we have to do. But yeah, I've had some I was teaching on a course in Belgium, like a week-long sort of microscope course for grad students and postdocs, sort of summer school. And I had to give a lecture. So I was really dumb of me. So I was supposed to give a lecture on a kind of microscope that I've been working on since my postdoc, so for like 15 years. And a friend of mine was also lecturing on the same summer school, and he's been doing it for like 20 years, and he's actually with his own hands built them.
Oh, wow.
So I suggested, so he was going to give, he was on the schedule to give a lecture on something else. So I suggested to the course organizer, well, maybe it's more appropriate for my friend to do this instead of me.
Mm-hmm.
And everyone said, oh yeah, that makes sense. Yeah, sure, that's great, good idea. And then it dawned on me that I've been giving lectures about that kind of microscope, and I have those already prepared.
Yeah.
But I've never given and the other topic was live cell imaging. So it's something that I'm totally into and happy, but I'd actually just never put together a lecture on live cell imaging.
Which takes a long time.
So then I was like, oh, this was great, except now I have to spend the next, month. So as part of that, there were certain things I wanted to, for example, illustrate how these fluorophores will bleach over time. So their ability to emit fluorescence will decrease if they're hit with too much
I
light.
remember that. I remember my professor teaching me back when I was doing this, it's like ironing. You have to constantly kind of move over the slide. You never want to let it sit in one place because it'll kind of burn a hole in the image.
It's particularly challenging with live cell imaging, though, because you are just, so there's always this balance between, how long you have your exposure set for, how often you take an exposure, and then, the total amount of light delivered and whether you can see the things you want to see. And if you, crank up the gain or whatever, it's going to get, the contrast is going to be blah, blah, blah. So I wanted to illustrate that. So I basically went onto a microscope and made a sort of, illustrative video of photo bleaching. And I did another one where if the microscope were to sort of bounce up and down and your thing goes out of focus. So I was able to go and just, play around for a while, which was a lot of fun.
It was fun. Yeah, that's really cool.
Also, we get to do demos for people, which is fun. Yeah, we had some, we had a PI that's in actually he's a faculty member in my same department. He had sort of some guests that came to visit his lab. He works on Alzheimer's and stuff, and his lab uses our facility a lot. So this was on a Saturday, and we sort of gave a tour, but as part of the tour, we did sort of these demos, and these people are not scientists at all.
Yeah.
And they're actually alumni from where he went to college. And so they were able to come in and we set up these demos, and it was really, really a lot of fun.
There's so much power, I think, in being able to provide a conduit, to provide a window to a world that we don't often see with our naked eyes. I think that's why telescope images are so powerful. I think that's why microscopy is so powerful, because we sort of evolved to think on a certain scale that's relevant to us. But obviously there's so many other scales in nature that unless we're directly faced with the data, we just don't think about it. We don't think about all the organisms living on our eyelashes all the time, We don't think about how tardigrades, for example, I point this way because I have a little tardigrade model over there that nobody can see because this is a podcast. But we don't think about how tardigrades are all over us all the time, and how we are made up of all these little individual cells, that even our cells are made up of all these other individual components. But you get to provide a window to that, which is like, I think, incredibly powerful.
Yeah, and in fact, one of the things I'm really interested in is and so people often conflate or confuse microscopist and microbiologist, and people often call me a microbiologist. And I'm like, no, I'm not.
No.
So the microbiology means that you study microbes,
Yeah.
which are bacteria, viruses, fungi, and but I am totally fascinated in them. I am absolutely eukaryotic mammalian cell biologist, but I'm still totally fascinated by other types of life, and it's like you said in the beginning, single-celled organisms. I mean, it's one thing to take a cell out of an organism and study it in isolation, and it's powerful. But there are also single-celled organisms. I've done a little bit of work. There's an algae that's called Chlamydomonas reinardi. It has nothing to do with chlamydia. It's called Chlamydomonas reinardi, and it is a photosynthetic single-celled algae that is actually motile. So it has these two little whip-like, sort of, I guess they're technically flagella, and they sort of beat and they swim around in water everywhere.
Yeah.
High elevation, desert, stream, whatever. And I did a little bit of work on them, and they're absolutely fascinating. But I'm really not a model organism type person.
When you study, let's say you're looking at human or, murine, like whatever, you're looking at mammalian cells from certain parts of the body. I'm trying to think of any time where these things would crash into one another. I guess gut, if you're looking at gut cells, or if you're looking at epithelial cells, or, sort of any place where there's, what do you call it? Like a membrane.
So a barrier, yeah.
Yeah, barrier, like eyes, nose, whatever. They're going to be swarming with microorganisms as well, right?
Well, hopefully not the ones that we're culturing.
Yeah, exactly.
So
that's a bad thing. Yeah,
that's the thing.
That's true, because you're looking at cell lines.
Exactly,
Yeah.
and they're generally cultured with penicillin and streptomycin in the media, and you try to keep them clean, and you don't want them to be contaminated, and when they are, you can usually tell, and then you throw them out.
Yeah, for a while, my master's research, I worked in a neuroscience lab. We were an electrophysiology lab. I had to learn all the ins and outs of electrophysiology, but I became very adept at doing cell culture, and I really started to enjoy that aspect of my job. So eventually I became the chief cell culture technician, and my master's thesis is actually about culturing specific fetal cells in certain parts of the mouse brain, substantia nigra, other places specifically for certain types of like Parkinson's model research. But I was fully into just culturing, feeding, taking care of, doing all the things you need to do to get these cells to be functional. And because we did pharmacology research, we weren't allowed to use antibiotics in our cell cultures. And of course, the brain has very little inherent immune response. So I remember the days of, the vent and the full, you got to get fully scrubbed in. And we even had a UV thing with a charcoal filter in the ceiling to try to sterilize the room. But still, you get fungal infections, you get bacterial infections, and it's torture. Yeah, you lose all this work. One little bacterial infection, and you're right.
Yep,
you literally throw it in the trash.
I think generally speaking, it's probably best to do the cultures without the antibiotics present, but
yeah, obviously different. If you're looking for physiological function, and you're adding drugs and you don't want to have drug interactions, that's one thing. But if you're looking at fixed cells, for example, it probably doesn't matter quite as much. But you're looking at these living cells and you're trying to see these biological processes within these living cells. I have to ask because, a lot of people, I didn't know. I was an undergrad in psych, and then I went into neurobiology. And I remember the first day that I took a neuroscience class, I knew I was in over my head because my professor said, neurons are just like any other cell in the body. They have a nucleus, a rough ER, a smooth ER, a mitochondria. And I was like, what are all these things you're oh, crap. I have to really step back and learn a lot about basic biology. And I assume that most people don't remember that from their high school bio classes. All the little subcomponents. What can you find in a cell? What does a ribosome do? What does, the Golgi apparatus do? What's your favorite component of a cell? Do you have one?
Well, actually, it's funny you say that because I had a very similar experience.
Really?
Because as an undergrad, I I went to Brown, and at Brown you can sort of you don't really have major requirements like most universities do. So I ended up taking all these classes, including a lot of independent study and research, with one professor, and he was a physiologist, and I learned all of this classic respiratory and metabolic physiology. And then I decided I wanted to go to grad school, and guess what? I needed to know biochemistry and cell biology and molecular biology and genetics and all that, and I had no idea. So it was a huge catch-up for me, too.
I love that. You're like sort of like secretly taking undergrad classes on the side to get ready.
No, I had to do some like summer school before I started my PhD.
Too,
absolutely. And then I had to really study really hard when I was doing the first year or two of my PhD.
Yeah, because those classes aren't easy. These upper-level undergrad biology classes are tough.
Yeah, absolutely. But what's my favorite component of the cell?
Is that a mean thing to ask?
No. So basically, the common theme to a lot of my research, going back to my postdoc, is the process of endocytosis. So that's how stuff gets into cells, right? So there are a few different ways that things can get into cells. Things can conceivably diffuse into them. So cells are all bound in a bilayer of lipids, okay? So it's this sort of, membrane that surrounds the cells. And things that dissolve in water or in aqueous environments won't generally just dissolve, just transfer across that membrane. It's a diffusion barrier. Yeah. So there can be, so you said you worked in an electrophysiology lab, so there are these things called ion channels. That's usually what you're studying if you're in an electrophysiology lab. And these are very, very specific pores, small tubes that exist in this membrane, and that will allow just sodium or just potassium to get through, for example. But if you're talking about like, lipoproteins, or you're talking about growth factors, or you're talking about, transferrin, which is the protein that binds to iron and floats around your blood. And then there's a thing called the transferrin receptor that cells have, and the transferrin carrying the iron binds to the transferrin receptor. Now, how does that receptor get into the cell so that the iron can be used by the cell? And the answer is endocytosis. And what it basically means is that a bit of this membrane bilayer is pinched off. It's sort of like gathered, like like a little beggar's purse, type thing, and basically pinched off. And there's this huge molecular machinery of tens of proteins that mediate this process.
These are like those G protein cascades.
So G protein.
Like, this was hard for me.
So G protein coupled receptors, can enter the cell through endocytosis. But the machinery that makes this happen, it's sort of, there are things that pinch the membrane in.
Oh, like the physical little molecular machines. Gotcha.
So you're basically, you have this like engineering problem of how you take a flat sheet of membrane and you invaginate it, make it into this little sphere that's then still attached by a little neck to the outside world, and then you sort of sever off that neck and then close it off and then move it into the cell.
It's so funny because I think people often don't, and we didn't often stop to think that these things are happening like your cells have machines in them, like actual, we call them molecular machines. And if you Google, like if you want to look, you can find GIFs or little videos of molecular machines that are, they're, what do you call it? Like models.
Yeah, animations.
Yeah, because these things are so small.
Absolutely,
but they're amazing. Like they walk, they unzip, they do all these cool things.
Exactly. So, but if you really want to, image the real things, then you get these super grainy pixelated images, more or less.
Yeah.
And that's sort of what we were doing, is we were imaging. We were among the first people to image the process of endocytosis event by event in live cells.
That's
amazing. So that's sort of how I started my postdoc. And so there's one protein in particular that's called clathrin. It's actually made up of two, there's what's called clathrin heavy chain and clathrin light chain. They come together. And basically what clathrin does is it forms these hexagonal or pentagonal structures. So really just like a little pentagon or hexagon, and then those stick together. And if you've ever looked at like a soccer ball, right, which is a combination of hexagons and pentagons that give it curvature. So you can actually make a sphere just by combining hexagons and pentagons, and that's exactly what happens inside the cell. So there's this one protein that comes together and forms these hexagons and pentagons that then coat these little pieces of membrane that are pinching off and sort of act like a shell around them or a cage around them. And it goes from a flat to a curved, depending on whether it's, just hexagons or hexagons plus pentagons. And that transition happens. It's just fascinating. So I'd have to say what we call the clathrin-coated vesicle would be the sort of thing inside the cell that I've just been the most fascinated with.
That's amazing. Isn't it cool how these things happen? Like there are these patterns in nature that recur all of the time that like a lot of times, a lot of new biological approaches are using what we call biomimicry, looking at the organism and the different ways that the organism moves and interacts, and then we're making new machines based on that. But isn't it cool how we oftentimes accidentally made things because we had smart people with a good grasp of mathematics and engineering who developed something like a soccer ball. And then now we find examples of it years later. I'm sure the soccer ball existed for years before we understood these clathrins. Did I say it right?
Yeah.
Okay, good. It's really cool to see that these patterns make sense at multiple levels.
Yeah, and like when RNA is made and the introns, the parts that are not coding, are spliced out, that's just like splicing out, from a tape, the dead air between the songs or whatever.
Absolutely. And that it's like sometimes the discovery came first and then we were able to use it to inform our engineering challenges. And sometimes, super inventive, incredible people did these engineering challenges. And then later we see examples.
So I don't know this. I haven't read the paper, but basically my wife showed it to me, but I haven't read it yet. But basically there's a paper that was just published where a movie, like an actual movie, was encoded into, I think, bacterial genomes.
Shut up.
Yeah, it's like it's very artistic actually. They picked a classical movie that's very, very old of like a horse running. It's like a couple of frames of a very silhouetted, because I saw the I haven't read the paper, but I saw the sort of, tagline type thing. And you see this and they basically encoded, using a bacterial genome, I think,
so cool.
a movie.
That's so crazy. Like you hear things about how like Craig Venter encoded his initials into things, or about how, do you remember when IBM did that movie where they were able to use an electron microscope to do like a boy and his atom, and they did a whole it's so cool to hear that this sort of art and science world of very, very small things are interacting in such a way that, and think about the capacity for data storage when we can encode things within bacteria or within nuclei of cells.
And I think there might be some people who would be sort of a little bit churlish about it and would say, oh, it's not that, it's not a really scientific, important application if you're just,
like it's a
carving
gimmick.
Your initials in atoms or whatever. But actually, I think it is really important. I think that, it's sort of, it's like planting the flag on the moon or whatever. Like it's really symbolic, not from a nationalistic point of view, but just from like there being a symbol, like a record, and having that be well thought out.
Oh, absolutely. It's like Juno. It's, I think, and I hope I'm not speaking out of turn. I don't think I'm incorrect when I say it's Juno, but maybe this was a Cassini thing. But I think it was a Juno thing, that it was an afterthought to add cameras, because there was like all of this data collection equipment on this
I remember hearing
flyby.
that.
Yeah, and it's like, oh no, maybe we should throw some cameras on so we can get some imaging. It's like, what captures the imagination of the public? Is it the data, or is it these stunning photos of storms on Jupiter? I mean, it's like so silly that we would even think not to do that, but sometimes those little afterthoughts, or sometimes these little, what we think of as being gimmicky.
Yeah. So one of the things is
matter.
So my wife works with Drosophila, with fruit flies, and Drosophila is famous because it's a genetic model organism, which means that you can make or just randomly make or specifically make or just identify mutants that then have some specific phenotype. And then the thing about Drosophila people is that they give them these completely kooky names, like Etheragogo is the name of a Drosophila mutant strain that under ether does some kind of thing, or
it go-goes.
Yeah, it go-goes. But they're just So if you're a Drosophila person and you make a mutant, it's like you have this, you have this responsibility to give it a totally cuckoo but really illustrative and sort of, dramatically artistic name. Whereas most mammalian cell biologists are just going to call it like, A2B13 is going to be the name of the mutant strain or whatever.
Although there is a protein called Sonic Hedgehog. Right?
Which I believe was originally a Drosophila protein.
There you go. That would be why. That's amazing.
Yeah, exactly.
Well, gosh, I know that you came to me while you're here visiting LA. We're here in person, and you are headed straight to the airport from our interview. So I don't want to keep you for too long. We've actually gone for like an hour and 15, but you're still fine because you were here early. I feel like there's so much more we could have talked about. Obviously, if you want to learn a lot more, guys, you need to read the book, The Cell. But before I let you go, I would love to ask you the same two questions I close all of my episodes with. You good with that?
Sure.
All right. So they're, the big picture questions. When you think about the future, in whatever context is relevant to you, number one, what is the thing that is, most disconcerting, that's keeping you up at night, that's worrying you? This could be a scientific problem. This could be a political problem. This could be a personal issue. What are you most concerned about? But on the flip side of that, to end on a positive note, what are you really, I think, optimistic, hopeful, excited about?
I mean, I think the answer to both is probably the same thing, actually, which would be automation. And it's especially relevant. I mean, this is a little bit self-serving or egotistical because, I think the question is, how much of science is going to be automated? And already, like, for example, we just got a grant from the National Institutes of Health to buy a robotic live cell imaging system, which can basically image 30 96-well plates in a cell culture incubator and do live cell imaging. And it can go from plate 1 to plate 30 and then back to plate 1 and go back within 1 micron or so of where the last image was taken because it's machined into such a high spec.
And it finds its own focus and stuff?
Yeah.
Yeah, that's amazing. Yeah. And so then you're like, am I out of a job?
Exactly.
Yeah.
Yeah. So, but not just, So I would say automation has so much potential, but then, I think there are a lot of people who are sort of, gets a little tinfoil hat. It goes to a little bit of an extreme when you have people, talking about like, the singularity and all that kind of stuff.
And the robots are going to take over. But it's true.
Skynet and all that. But
if you work a manufacturing job, you may have seen that less and less people hold the same position because there are so many great manufacturing robots and machines that can do the work of, X amount of people. Like, I think this is an interesting and important concept to explore simply because it runs the gamut from these incredibly high-skilled technical jobs where you have to have a PhD and a postdoc and years of research to do it, all the way to a position on a factory line at, a GM plant where these cars are being made by machines.
And I think that, when you get, obviously this is not my area, but when you start to talk manufacturing or other areas and you start to talk about efficiency and productivity, it's like for the benefit of whom, right? Is it so that more profit can be made, or is it so there can be more production
for the consumer?
Exactly.
Or is it about and really I think it goes back to these arguments that we often see, not to get too political, where we see Trump, for example, bending over backward for coal miners' jobs, where there's this conversation about, well, how many coal miners are there actually in this country anymore? And who really, we're talking about the benefit of these individual workers, which of course nobody is going to question that these individual workers shouldn't be able to have access to a well-paying job and benefits and things like that. It breaks my heart to see people being phased out of their work. But at the same time, for the greater good, is mining coal really the direction that we should be going?
And if you look at like the last, I think since World War II, and you look at farms, for example, the American food production and consumption system, right, over the last 60 years or so, right, has become completely automated, and yes, it can create lots of calories very cheaply, but to what end? The family farms have been put out of business, and the people who are consuming those calories are still malnourished because it's not all about calories, right?
Absolutely. Like, don't even get me started on corn subsidies. Yeah.
So
And the fact that
exactly.
all of our livestock is eating corn, and we're eating high fructose corn syrup and everything.
So the past few decades would suggest that it's, probably not going to go so well.
Yeah, it's true. It's interesting. And I know that there are authors who have focused on this. And you're right. Sometimes it goes to this crazy, like, Ray Kurzweil extreme of the singularity and, like, this intense futurism. But that sweet spot in the middle, I think, is more than just a philosophical problem. It's a practical problem. That's an interesting answer. I don't know if anybody on the show has answered that way before, especially coming from a cell biologist. I love that. Well, Josh, this is so much fun. I'm really excited to be able to share the book with everybody. I think, especially for my audience here on Talk Nerdy, this is right up their alley. So you guys, you should reach out to us. You should let us know if you got the book and what you think. Again, it's The Cell, Discovering the Microscopic World That Determines Our Health, Our Consciousness, and Our Future. And it's in paperback now. Yep. Yeah, so you can pick that up. I'll link to it on the show. Josh, let everybody know where they can find you. Are you active in social media if they want to say hi, if they want to say they checked it out?
I'm really not so active in social media. But the facility that I run, the Center for Advanced Microscopy and Nikon Imaging Center at Northwestern University, Feinberg School of Medicine, we've got our own website.
Very cool.
I have a page on the publisher is called Ben Bella, and so if you Google me and Ben Bella, or just Google me and The Cell, you can get that paging. People can contact me through the publisher, I think.
Awesome, and I'll link out to all of that so that it's easy for you guys. Well, Josh, thank you once again for joining me
Thank
today.
you so much. It was really great.
It was awesome. And everybody listening, thank you for coming back week after week. I'm really looking forward to the next time we all get together to talk nerdy.